Hi guys, welcome to Learn with Gogs. இன்னைக்கு வீடியோக்குள்ளே போகிறதுக்கு முன்னாடி ஒரு ஆட் பார்த்துடலாம். BuyMote ஷாப்பிங் ஆப். இது ஒரு ஆன்லைன் ஷாப்பிங் ஆப். இதில் க்ராசரிஸ் எல்லாம் அவைலபிளாக இருக்குது. மற்ற ஆப்ஸோட கம்பேர் பண்ணும்போது இதில் குவாலிட்டியும் அதிகமாக இருக்கும், அதே மாதிரி டிஸ்கவுண்ட்டும் கிடைக்கும். ஸோ ப்ளே ஸ்டோர் அண்ட் ஆப் ஸ்டோர் ரெண்டுலேயுமே இது அவைலபிளாக இருக்குது. டிஸ்கிரிப்ஷனில் லிங்க்கும் டீட்டெயில்ஸும் இருக்குது. செக் பண்ணிவிட்டு இன்ஸ்டால் பண்ணி பை பண்ணிக்கோங்க. இது ஒரு ரொம்ப யூஸ்ஃபுல்லான ஒரு ஆப். அண்ட் இதில் ஒரு இம்பார்ட்டண்ட் விஷயம் என்னென்னா டோர் ஸ்டெப் டெலிவரியும் அவைலபிளாக இருக்குது. ஸோ மிஸ் பண்ணிடாதீங்க, யூஸ் பண்ணிக்கோங்க. ஸோ டுடே we are going to see about chromosomal DNA isolation. That is, why we have to isolate the DNA from the chromosome, because to study the genetic characters of the organism, if the organism is sometimes pathogenic, so we have to extract the DNA is important to study the genetic causes of the disease and for the development of diagnostic and drugs. So this is a main thing. We have to isolate the chromosome DNA, okay? And the materials required for this chromosomal DNA isolations is TE buffer. We have to make up to 10 ml. Here 10 ml is required, so we are make up to 10 ml. pH should be maintained at 8, okay? Here we have to add 10 millimolar tris HCl and 1 millimolar EDTA. These concentrations and measurements will be laterly I'll told to you. Second, we have to take E. coli culture which will inoculated in LB broth. We have to take the 24 hours culture, which is mainly important here, and we have to take 10% SDS solution. Here also we have to make up to 10 ml. That means 1 gram in 10 ml distilled water, okay? Next thing, we have to take proteinase K, and we need phenol, chloroform and isoamyl alcohol in a ratio, which means 25 ml phenol, 24 ml chloroform and 1 ml isoamyl alcohol. So we can make it up to 50 ml, okay? Then we need sodium acetate. Here we have to need this in 3 molar concentration and maintain the pH 5.2. I'll tell this later to you. Then 70% ice cold ethanol we needed. 70% means for 100 ml take 70 ml of ethanol and 30 ml of distilled water. Mix it, we can get 70 ml of ethanol, and we have to place in ice cold container or deep freezer. Then agarose gel electrophoresis we have to run finally to see the bands are separated from the samples, that is DNA separated from the sample. Here 2% agarose we have to taken. That means 2 gram agarose in 100 ml of TAE buffer we have to mix. Here we only need 50 ml, so 1 gram of agarose we have to take and distilled in diluted in 50 ml of TAE buffer, not in distilled water, okay? Here we have to prepare tank buffer, that is tris acetate ADTA TAE buffer in 1x concentration for 1000 ml. Then the principle of this chromosomal DNA isolation is we need to identify the presence of organism in a specific test. This is the main principle, and I'll tell the all reagents purpose in this, okay? LB broth which contains high nutrients, so we specifically use this LB broth in this test. This LB broth contains tryptophan, yeast and NaCl. 16 to 18 hours culture is highly required to isolate the DNA, okay? And SDS solution acts as detergent. It can denature and disturb the bacterial cell membrane, so this detergent is used, okay? Then phenol, chloroform and isoamyl alcohol. Here phenol helps to precipitate the protein while centrifugation, and this phenol helps to get the pellet, okay? And chloroforms, this chloroform makes 2 layers, that is organic phase and aqueous phase. Bottom of this tube, effluent of tube is organic phase, and the upper layer supernatant part is aqueous phase. And isoamyl alcohol, it is used to avoid foams during certification, because we added SDS here. It is the detergent. While adding detergent, absolutely foams will be formed. To avoid those foams, we have to add isoamyl alcohol to control that, okay? And we have to add sodium acetate 3 molar concentration. This will help to neutralize the DNA, because DNA is negatively charged, so that can break down easily. So we have to make it neutral, and the DNA is safe during isolation. Okay, and tris HCl and EDTA are the components of TE buffer. This tris HCl maintains pH and EDTA to protect DNA. This EDTA will bind to the DNA's enzyme, which is present inside the DNA or chromosome. The DNA's enzyme will present. That enzyme can denature or defect or make any cut in the DNA. So here this EDTA will go and bind to this DNA's or RNA's enzymes and prevent the denature of the DNA or RNA while isolating. That why we have to add the EDTA, okay, so that not able to affect the DNA, this enzyme. And 70% ethanol will precipitate the DNA, okay. And agarose gel, this agarose gel is helpful to different the lengths of DNA by running it into the gel. And while running the electrophoresis, this TAE buffer is called tank buffer. This is mainly used to all electrophoresis unit because it is used to run electrophoresis, which is carries current and maintain pH of the medium. Here current will be supplied, the samples are loaded, is must be present at the negative side of the electrophoresis unit. When negative charge applied and those DNAs, those samples will be contains negative charge. When it will run through positive towards the positive side, then we have to isolate the long fragment or small fragment of DNA. Okay, so next one is procedure. Here we have to take the Eppendorf tube, okay, then add our E. coli culture, 18 to 24 hours E. coli culture, 1.5 ml, add it in the Eppendorf, then centrifuge the Eppendorf tube containing culture in 13,000 or 10,000 rpm. Always we should use high speed rpm to isolate the DNA from the cells, okay. And collect the pellet after centrifugation. Yes, after centrifugation pellet is formed under the Eppendorf tube bottom of the side. We have to collect the pellet and discard the supernatant. Then with that pellet add TE buffer 200 ml. Using micropipette we have to add 200 ml TE buffer and add 400 ml 10% SDS solution, okay. Then here we have to incubate this solution with 65 degrees Celsius for 10 minutes. That means we have to kept it in boiling water bath for 10 minutes, okay. Then after 10 minutes we have to add proteinase K, that is 60 ml. The measurement of proteinase K is 1 ml distilled water we have to add 20 mg, okay. So 60 ml we need means we have to add 3 ml distilled water and we get 60 ml proteinase K, and we have to add 60 ml proteinase K in that Eppendorf and incubate at 37 degrees Celsius for 1 to 2 hours, okay. After that we have to add equal volume of, that means these three chemical reagents, phenol, chloroform and isoamyl alcohol, in the ratio I told you already. That ratio we have to add 660 ml, and we have to mix it by invert the tube because vigorous mixing will damage the DNA present inside the cell, okay. Then after that we have to centrifuge at high speed for 5 minutes. Two phases will separated, like I already told you, aqueous phase and organic phase. Here transfer the aqueous phase only, I mean this supernatant phase. Here we need that supernatant phase only, so transfer the aqueous phase into a fresh tube and again repeat the same steps 3. Here without phenol, chloroform and isoamyl alcohol only we have to add and centrifuge at maximum speed for 5 minutes. After that again aqueous solution is transferred to new tube. Add 100 ml of 3 molar sodium acetate, add 1 ml of ice cold ethanol, I mean that 70% ethanol I told you before, and incubate for 1 hour. After that, again we have to centrifuge for 10 minutes, and now we will have clear pellet, thread like structure pellet in the bottom of the Eppendorf tube. And now discard all the supernatant and kept only the pellet. In that pellet we have to add 50 to 100 ml of TE buffer again and kept it as a sample now. Okay, then we have to prepare this that agarose gel I told you, 1 gram in 50 ml, and then we have to ready TAE buffer and pour it into the tank and load the samples in each well. I'll show you the result now. This is the result. Here this line indicates the separation of DNA from the cells. This will not separation occurs. Here DNA will not separated from the cells, and these samples are failed, and this will give correct result. Okay, here the DNA will separate, and here the band, the fragment, the pieces of DNA is round. Okay, so this is a correct step, we get the result. So you will definitely get the result by proper usage of these procedures and the preparation of the correct amount of reagents. Okay, here if you want this calculation, this is a tank buffer, that is TAE and TB buffer calculations are here. You have to check it if you want. And here the sodium acetate 3 molar concentration calculation is also here. Thank you for watching. If you like this video, please hit the like button and subscribe my channel for more videos. Thank you.